Good morning to all the students. Welcome to biology session today. Today we are going to discuss the chapter plant growth and development. From plant growth and development, what we are going to discuss today is the types of seed germination and what are the factors that affect the plant growth, and then we are going to learn in detail about phytohormones and their physiological functions and applications. First, let us look into the types of seed germination. Seed germination is of two types. One is epigeal seed germination and second is hypogeal seed germination. In epigeal seed germination, what happens is the cotyledons will rise above the ground and first hypocotyle will emerge from the seed. Okay, it is first hypocotyle that emerges from the seed and it is first radical that grows. Okay, in epigeal seed germination, first hypocotyle emerges and radical elongates and cotyledons will come above the ground. Whereas in hypogeal seed germination, it is completely vice versa. Okay, it is first epicotyle that emerges, and later radical will develop. Okay, later radical will develop. First epicotyle emerges and later radical will develop, and cotyledons will remain underground itself. Okay, cotyledons remain underground itself. Best example here I can take is groundnut, which is Arachis hypogea. So the basic differences between epigeal and hypogeal seed germination. Epigeal seed germination where cotyledons will rise above the ground and it is first hypocotyle that emerges from the seed. Whereas hypogeal seed germination, cotyledons will remain underground throughout the period of germination and it is first what emerges first? Epicotyle emerges first. Got it, ma? So these are the two types of seed germination. And then what are the growth factors that affect the growth of a plant from the time of seed germination to the period of senescence? What is the growth of plant driven by? Okay, it is driven by growth factors, both internal and external growth factors. Internal growth factors are also known as intrinsic growth factors. External growth factors are also known as extrinsic growth factors. So internal or intrinsic growth factors can be of two types. First type is intracellular or genetic factors. Second type can be intercellular factors. Those are chemicals such as plant growth regulators. I will give you a brief introduction on what are plant growth regulators. So here there are two intrinsic factors which govern the development of plant body. One is intracellular genetic factors, second is intercellular factors like PGRs, plant growth regulators, which are also addressed as plant hormones or phytohormones. Plant hormones or phytohormones. Second is extrinsic growth factors or external growth factors. Can be light, temperature, water, oxygen and nutrients. These are the extrinsic growth factors. First, if you have to learn in detail about plant growth regulators, plant growth regulators are also addressed as phytohormones or plant hormones. What are these known as? These are small chemical substances, small organic chemical substances that are secreted in any part of the plant body and they will reach to the place of action and perform the action, be it growth inhibition or growth promotion or modification inside the plant. Okay. So what are plant growth regulators? They are small organic chemical substances secreted or synthesized in any part of the plant body which either promote the growth of the plant or inhibit the growth of the plant or modify the plant body. Okay. So these are small organic chemical substances synthesized in any part of the plant okay. They can promote the activity of inhibition, they can inhibit the growth of the plant or promote the growth of the plant, or they are also involved in modification of plant body, modification of plant body okay. These are what plant growth regulators, and this is the diversity of their role. What are the 5 types of plant growth regulators that we are going to learn? So, first, as we have discussed, they can either promote or inhibit the growth of the plant. Based on their functional growth, plant growth regulators are classified into plant growth promoters and plant growth inhibitors. Yeah, they are classified into plant growth promoters and plant growth inhibitors. Plant growth promoters and plant growth inhibitors. Plant growth promoters include auxin, cytokinin and gibberellins. Auxins, cytokinins and gibberellins. These phytohormones will promote the growth of the plant, be it in the form of cell division, cell elongation, increase in the length of the plant. Okay, this will promote the growth of the plant. And plant growth inhibitor includes only one phytohormone, which is also known as stress hormone or anti-transparent hormone, that is abscisic acid. Okay, and ethylene is that phytohormone, ethylene, which performs the dual role both as plant growth promoter and as plant growth inhibitor. Clear with this? So, plant growth promoters include auxins, cytokinin and gibberellins. Plant growth inhibitor includes abscisic acid, which is also known as stress hormone, because it will be released or secreted when the plant is in stress, be it heat stress, water stress. Okay? And ethylene is the hormone which performs dual function both as plant growth promoter and as plant growth inhibitor. So, today first we are going to learn about only the functions, physiological functions and the applications of the hormone. Many a times you got the NEET questions only on the functions, physiological functions and applications of any phytohormone. So, first we are going to learn about auxin. Before learning about auxin's physiological function, you have to know about two things. Auxins, we have two types of auxins. One are naturally secreted from the plant body, second are synthetically made. Okay? So, as such we have two types. Auxins are of two types. One is natural auxins, second synthetic auxins. Synthetic auxins. Natural auxins include indole acetic acid and indole butyric acid. Synthetic acids, synthetic auxins include naphthalene acetic acid, 2,4-dichlorophenoxyacetic acid. These are the synthetic auxins. Remember both the natural auxins and synthetic auxins, because these are very much essential to remember the applications of auxins. First, know about the physiological functions of auxins. First is apical dominance. Quite a times you got in NEET and MCET. Okay, apical dominance is very important. What is apical dominance? If I have to consider this as the main apical part of the plant, and these are the lateral buds these are the lateral buds and this is the apical bud. Okay. Auxins are majorly present in the shoot apical meristem and root apical meristem. Thereby, auxins will promote the growth of the apical buds. Okay, auxins, they will promote the growth of apical buds and they will inhibit the growth of lateral buds. This phenomena is known as apical dominance. Okay, auxins present in the shoot apical meristem or root apical meristematic regions, they will promote the growth of the apical buds and inhibit the growth of lateral buds, that is apical dominance. Second is cell division. Okay, auxins will promote the cell division, thereby increase in cell number happens and this plays a major role in formation of primary plant body. Next is abscission. Okay, what is abscission? Fall of older leaves and ripened fruits from the plant is known as abscission. Auxins will promote the abscission of older leaves and ripened fruits, and auxins will inhibit the abscission of younger leaves and buds. Okay, it plays dual function for abscission. It will promote the abscission of older leaves and ripened fruits and it will inhibit the abscission of younger leaves and buds. And auxin plays a major role in xylem differentiation. Okay, the concept of differentiation, dedifferentiation, redifferentiation, you will learn it in anatomy chapter of NCERT 11. But as of now, you remember that auxin also promotes the differentiation of xylem tissue. So, 4 major functions of auxins ma. First is apical dominance, second is cell division, third abscission and fourth xylem differentiation. How are we going to apply these auxins for the growth of plants? Okay, applications of auxins. First is rooting. Auxin is said to be a rooting hormone. It is because auxin promotes rooting. Okay, in tissue culture labs and all, auxins are induced to promote the rooting. Second, auxins will promote flowering. Okay, rooting, this is promoted by naphthalene acetic acid and indole butyric acid. Okay, flowering is promoted by naphthalene acetic acid and 2,4-dichlorophenoxy acetic acid. And then parthenocarpy. What is parthenocarpy? Production of fruits without fertilization or formation of fruits without fertilization is known as parthenocarpy. So, seedless fruits are produced by the application of auxins, and what type of auxins we will apply ma? Indole acetic acid and indole butyric acid. Okay. So, what is parthenocarpy? Formation of fruits without seeds, that is production of seedless fruits. Next is weedicides. Okay, weedicides, auxins, some synthetic auxins like 2,4-dichlorophenoxy acetic acid. Okay, they are used to produce weed free lawns. Okay, application of 2,4-dichlorophenoxy acetic acid will help in formation of weed free lawns. Clear right till here? So, these are the functions. First you have to know types of auxins, functions, and these are the applications of auxins. Second point you have to remember is the discovery part, precursors and derivatives of auxins, which are the major part of every phytohormone, that you will be learning in the NCERT 11. As of now, remember what are plant growth promoters, plant growth inhibitors, and what are the functions and applications of each phytohormone. Second phytohormone that we are going to exercise is cytokinin. Okay, cytokinin. Cytokinins. Okay, the major function of cytokinins is cell division. Okay, now you might be asking me, ma'am, auxins also perform the activity of cell division. Cytokinins also perform the activity of cell division. For example, if we got a question that which phytohormone will promote cell division and 4 options are given, amongst that one will be auxin, one will be cytokinin, what option we need to choose? For this you should know about 2 phenomena, one is synergistic action, second is antagonistic action. 2 or more hormones performing only one function, single function, is known as synergistic action. Okay? Synergistic action. Here 2 or more phytohormones, they will perform only single function. Second is antagonistic action. Antagonistic action. Okay, antagonistic action. Here one hormone will promote the activity, other hormone will inhibit the activity. For example, cytokinin will delay the senescence. As it promotes the cell division, it will increase the amount of fresh cells or new cells, whereby it delays the process of senescence, which is proved by Richmond-Lang effect. Okay? So, cytokinin will delay the process of senescence, whereas abscisic acid, okay, abscisic acid will promote the activity of senescence. So, these 2 phytohormones are antagonistic for a single function. Okay, like that we have synergistic hormones or antagonistic hormones. With respect to cell division, auxins and cytokinins are synergistic in their role, but of them more intense cell division is performed by the hormone auxins, and mostly cell elongation or increase in cell number and cell elongation will be performed by auxins, but more intensely cell division is relayed on the hormone cytokinins. Okay? So, auxins and cytokinins are synergistic in their action when we take cell division into consideration. Second is senescence. Cytokinins will delay the process of senescence, abscisic acid will promote the senescence. Next, auxins will overcome the apical dominance. Now, with respect to apical dominance, auxins will promote the apical dominance. Auxins will promote the apical dominance. That is, auxins will promote the growth of apical bud and inhibit the growth of lateral buds. But if you apply cytokinins, the apical dominance phenomena will be reversed, because cytokinins will help in the growth of lateral buds, whereby apical bud growth will be inhibited. Okay? So, cytokinins will counteract the activity of what? Auxins with respect to apical dominance. Fine, right? So, 2 antagonistic hormones with respect to apical dominance is auxins and cytokinins. 2 antagonistic hormones with respect to senescence is cytokinin and ABA. You have to remember this. Okay? And then growth of shoot and leaves. Okay? Increase in lateral branches or lateral shoots and increase in the number of leaves is by cytokinin itself. Now, applications of cytokinin are majorly used in tissue culture. This will be along with auxins. Remember, if auxins and cytokinins are in equal ratio 1:1, then this promotes the formation of callus. Callus is undifferentiated plant mass, and if auxin concentration is more and cytokinin concentration is less, auxin concentration is more and cytokinin concentration is less, this performs the activity of rooting. Okay. Next, if auxin concentration is less and cytokinin concentration is more, this performs the activity of shooting. Okay. So, auxin cytokinin ratio, if it is same 1:1, this will help in the formation of callus. Auxin concentration is more, cytokinin concentration is less, this promotes rooting. Auxin concentration is less, cytokinin concentration is more, this promotes shooting. Got it, ma? These are the applications of what? Cytokinins in tissue culture. Next, about gibberellic acid. or gibberellins. Gibberellins will promote the seed germination. Okay, gibberellins will promote the seed germination. This I will explain you very clearly by taking an example of monocot seed. Let me take an example of maize seed. So here we know maize seeds will have endosperm. It has two types of endosperm. One is proteinaceous layer, that is aileron layer. Second is starchy endosperm. Starchy endosperm. Fine, right? So the starch will be simplified by the action of what, ma? Amylase. Starch will be simplified by the action of amylase. Amylase will be produced by the induction of gibberellic acid. Gibberellic acid will trigger the release of the enzyme amylase. Amylase will solubilize the starch and simplest sugars will be transmitted to the embryonal axis, wherein towards the tip it will grow to plumule, and over the plumule there will be a covering sheet called as coleoptile, and below it will be growing into radicle, which is covered by a protective covering, that is coleorhiza. Got it? So plumule is covered by coleoptile, radicle is enclosed by coleorhiza. Got it, ma? So here what is the role of gibberellic acid? To promote or induce the release of amylases. Amylases will solubilize the starch. Simplest sugars are transferred to the embryonal axis, which will help in the seed germination. It will help in seed germination. Okay. So gibberellins will promote seed germination. Second is bolting. What is bolting? Especially in the rosette plants. In rosette plants, for example, beetroot, cabbage, cauliflower. Okay. In such plants what happens is, gibberellins are sprayed prior to flowering, so that this increases the internodal elongation. Once the internodes are completely elongated, post fertilization, this forms large sized vegetables or fruits. Okay. Gibberellins are also sprayed over the fruits like apples, grapes, so that when the stalk length is increased, fruit size will be increased, and you will get many grapes as the fruitlets when the main stalk length is increased. So gibberellins are sprayed just prior to the flowering, so post fertilization you will get large sized fruits and vegetables. Okay. So gibberellins are sprayed prior to flowering. Got it? Next is stem elongation. Okay. Gibberellins will increase the internodal length of the stem, whereby this is highly used for increasing sugarcane yield. Increasing sugarcane yield. Got it? So gibberellins are highly used for internodal elongation, which is used to promote the sugarcane yield. These are the three main functions of gibberellins. One is seed germination, second is bolting, third is stem elongation. And then gibberellins, as they help in the secretion of amylase, they are highly used in brewing industries for malting process. For malting process. Next, this will enhance the sugarcane yield. This will promote quicker maturity in conifers. This will promote quicker maturity in conifers. This will delay ripening, and this will promote the fruit growth, wherein the stalk size increases and the fruit size also increases. These are the functions and applications of gibberellins. And then next is abscisic acid. abscisic acid, which is also addressed as stress hormone because it will be released during the stress conditions. This is also known as stress hormone or anti-transparent hormone. Okay. Abscisic acid is known as stress hormone or anti-transparent hormone. This will promote the stomatal closure, auxins. For example, there is heat stress or there is high temperature conditions. To prevent the process of transpiration, the immediate response a plant shows is closing the gates. What is its gate? Stomata. So, immediately stomata will be closed. This stomatal closure is triggered by the release of abscisic acid. Second is abscission. We have learned when we are learning the functions of auxins that auxins will promote the abscission of older leaves and ripened fruits, and it will inhibit the abscission of younger leaves and buds. Whereas here abscisic acid, it will promote the fall of buds and younger leaves as well. So, with respect to abscission, which two hormones are antagonistic to each other? With respect to abscission, auxins and ABA are antagonistic to each other. Remember that. Next is seed dormancy. During unfavorable conditions, the seeds will relay in the dormant stage. That is because of abscisic acid. It will not allow the seed germination during unfavorable conditions. So, with respect to seed germination and seed dormancy is promoted by abscisic acid and inhibited by gibberellic acid. To put it in more proper terms, seed germination, I can say the function seed germination is promoted by gibberellic acid and inhibited by abscisic acid. So, these two hormones are antagonistic to each other with respect to with respect to seed germination. These two hormones are antagonistic to each other, and abscisic acid will inhibit the metabolism, whereas cytokinins will promote the nutrient metabolism. Cytokinins will promote the nutrient metabolism, whereas abscisic acid will inhibit the metabolism. So, when it comes to nutrient metabolism, which two hormones are antagonistic to each other? Cytokinins and ABA. Always do remember, students, synergistic and antagonistic functions are very important. Firstly, you have to know the functions and applications of all the functions and applications of all the phytohormones, and then you have to look into the synergistic and antagonistic functions as well. And then applications of abscisic acid. First is dormancy. It will promote the seed dormancy during unfavorable conditions, and this acts as anti-transparent hormone as well. Okay, so these are the functions and applications of auxin. The last phytohormone that we are going to look into is ethylene. Functions and applications of ethylene. First, functions of ethylene. Ethylene promotes flowering, and the major function of ethylene is fruit ripening. It gives triple response. It promotes senescence. Ethylene promotes senescence and it helps in the formation of adventitious roots. These are the functions of ethylene. And ethylene in the form of ethaphon. Ethylene in the form of ethaphon is applied for fruit ripening. This promotes abscission and thinning of the fruit wall at the time of ripening. This will increase the number of fruit set. Okay, it will increase in fruit set, and sprouting of storage organs also happen. This happens when ethylene is applied to the plant organ. So, these are the functions and applications of ethylene. Now, we will look into few of the NEET PYQs where the questions are being asked from phytohormones. First question, which one of the following phytohormones will promote the nutrient mobilization which helps in the delay of leaf senescence in plants? Nutrient mobilization is promoted by cytokinin and it is inhibited by what? Abscisic acid. Very simple and direct question from the NCERT. Next question, this is from Auxins. This is from the phytohormone auxins. Auxin is used by gardeners. Auxin is used by gardeners to prepare weed-free lawns. Okay, and which auxin is applied? Which type of synthetic auxin? 2,4-dichlorophenoxyacetic acid. But there is no damage is caused to grass as auxin. Promotes apical dominance, promotes abscission of mature leaves only. They do not affect monocotyledonous plants. They can help in cell division in grasses to produce growth. Okay. So, the right answer will be option 3 because 2,4-D is applied to eliminate dicot weeds. Dicot weeds. Next. This question you look into, ma, asked in the year NEET 2024. Yeah. Spraying sugarcane crop with which of the following plant growth regulators will increase the length of stem, thus this increases the sugarcane yield. The right answer will be Gibberellins. Okay. By spraying Gibberellins, internodal length will be increased whereby sugarcane crop yield will be increased. Fine, right? And then this is the last question of today's session which is asked in the year NEET 2023. Spraying of which of the following phytohormone on juvenile conifers will help in hastening the maturity period that leads to early seed production. This leads to early seed production. Which phytohormone? Of course, it is cytokinin, and the natural form of cytokinin is zeatin. So, the right answer will be option 3. Fine, right? So, with this, the concept of phytohormones is exercised in detail. The key takeaways of today's session: at the beginning of the session we have learned about epigeal seed germination and hypogeal seed germination, followed by factors that promote factors that affect the growth of a plant, both intrinsic and extrinsic factor. One of the intrinsic factor is plant growth regulators. We have two types of plant growth regulators. One is plant growth promoters, second is plant growth inhibitors. Plant growth promoters include 3 sets of hormones. One is auxin, gibberellins and cytokinins. Plant growth inhibitors include abscisic acid. Ethylene will play dual role. And then we have looked into the functions and applications of each phytohormone. At last we have exercised few PYQs also. So, in today's session we got an idea on phytohormones. Later when we are going to NCERT 11, we have to learn each phytohormone in detail by knowing the discovery part, precursor, derivatives, functions and applications. Got it, ma? Thank you.